Hello, my name is Victor Zberstis, and I'm on the team working behind the scenes on WorldCommunityGrid.org, which has been harvesting spare processing time from computers around the world since 2004, putting those computers to good use while they would otherwise be consuming electrical power just waiting for that next keystroke or mouse movement. One of the research projects on World Community Grid, the Help Conquer Cancer project from the Ontario Cancer Institute in Toronto, Canada, has just completed the grid computing portion of the project. Please let me introduce Dr. Igor Jurisica, who will tell you what the Help Conquer Cancer project has accomplished so far and what their plans are for using the results of the computations completed on worldcommunitygrid.org.
Thank you, Victors. Welcome, everybody. It's very exciting time to finish the World Community Grid computation that we started in 2007 in November. This is only one of the many areas that we focus on trying to identify what causes cancer and how can we help individual patients. This first slide really highlights diversity of applications and programs that we are working on. Some of them, for example, those blue and red curves on the top middle part of the graph identify patients that have a better or worse prognosis, and through those we can identify better approaches for the treatment. The big blob network in the middle of the graph is protein interaction network, and we need to understand how these individual proteins function together, how they create pathways, how they are deregulated under disease conditions, so we can better identify the treatments. Which links us to the images that are on the right side of the screen, and those are individual molecules or compounds. They are basically drugs used for treatment of cancer and other diseases. But this specific presentation and this specific project was on trying to identify better methods for determining three-dimensional structure of the proteins. And those are the few images that you see on the bottom of the screen. Cancer is a devastating disease, and trying to solve some of the aspects of cancer on the World Community Grid is very fitting because it affects millions of people around the globe. And now we are using donated computing power around the globe to help us understand protein crystallization process in more detail, which is necessary for understanding function of the proteins that are related to this disease. As you can see in the statistics on this slide, there are some cancers in which we, meaning global we, researchers around the world, reasonable progress. Prostate and breast cancer, most of the patients do have good long-term survival. Unfortunately, cancers like pancreas still have very short survival for most of the patients. This disease affects all of the nations around the world, and we need to combine our resources in order to try to solve it. These curves highlight, that you can see on the right side, highlight that if we identify disease early, this is a stage 1 disease, green curve, most of the patients will have a good survival long term. Unfortunately, as the red curve and stage 4 and stage 3 disease highlight, if we identify cancer late, we cannot help most of the patients. And so what we try to do on a computational side is to identify some biomarkers, be it gene, proteins, microRNAs, or other entities that can help us identify disease earlier. The other very strong part where computational science can help significantly is to identify groups of patients and identify patients that are at a higher risk, and thus we may need stronger, more aggressive treatment, and also identify groups that might not respond to the current treatment and thus may need some alternative strategies. The last part really is that we not only want a clinical tool, kind of a black box, which would be definitely useful, but we also want to understand the mechanism so we can hopefully improve ways of understanding how does it start, why does it start, can we stop it by potentially using novel medicine. So many of you probably remember Tetris game, and there are two-dimensional versions where the task is really to lay individual bricks of different shape in a most effective way. The challenges really start to be interesting when you move into three-dimensional Tetris, because now visualizing in all three axes how to best place individual bricks is far more complicated. And that's precisely that brings us to how understanding the protein and their structure can help us understand how the disease starts or how can we treat it. This example, the big gray, red, and blue blob in a bag is a three-dimensional structure of the protein MDM2. And the little diagram in the front is a drug called Nutlin that basically modifies function of this protein. And thus, we need to understand the structure in order to be able to design novel medicines and also help individual patients. World Community Grid is a global effort. We benefited significantly from this massive computing resource in order to help us understand the protein crystallization process better. But also in return, we create various other resources from other analyses and other programs that can be beneficial to other researchers. So we make them available on our portals, and we make available both the data, results, and also the programs. These are just some of the few examples from our lab. The data that we have on this molecular data that we have on a complex disease like cancer spans many different modalities, and it's really highly dimensional. Tens of thousands of variables for individual data sets for each patient sample. So we need to combine multiple resources, different tools and algorithms in order to be able to help us understand and interpret what does it mean when cancer starts. I touch upon a little bit on the signatures, which is trying to identify which patient groups have a better prognosis or worse prognosis, which patients may or may not respond to current treatment strategies, and so on. I also showed one of the examples for the networks, because networks enable us to connect these multiple different pieces of information and have a better visualization and interpretation of it. But really, the Help Conquer Cancer project on a World Community Grid now was about the structure determination and protein structure, specifically X-ray crystallography approach, which are the images that you saw on your screen many times. The process and this integrative analysis obviously doesn't happen overnight. It takes many years. But eventually, there are interesting outcomes that will start hopefully changing lives of cancer patients. One such example that happened over the course of this project, although not directly related to it, is Lung Cancer Express DX, which is a prognostic signature for lung cancer patients, which is now being brought to market by Precision Diagnostic from Pittsburgh. So, some background on X-ray crystallography. While there are many different approaches for determining structure, X-ray crystallography is de facto standard. And as you can see on the bottom pie chart, most of the structures currently are determined by X-ray crystallography in protein structure initiatives. So there are other methods, but this method determines most structures for most proteins. As you can see on a bar chart just above it, the problem we are facing is that the task is very complicated. First, we have to clone the protein, then it has to be expressed, purified, and so on. And as you can see, this dropping curve and the bar chart basically identifies that for all of the proteins that we are interested in determining, very small fraction will have structure determined through the course. And the problem really starts from the fact that nature is complicated, and the more disease-related protein or the more interesting the protein is, the more difficult it is to crystallize. So in order to help with this process, colleagues from Hauptmann Woodward Medical Research Institute in Buffalo started an automated process for screening a large number of different conditions that can lead to crystallization. And thus they turned this chemistry and protein crystallography problem into an information technology problem, because now we have vast amount of data coming from these experiments. And that's where my team with Christian Kamba and all of the others started to look into how can we harness this information for improving the process. So you saw many of the images going through your screen during the process of analyzing these images from the crystallography, but you may have wondered where they are coming from. So after initial experiments, basically large pipetting robots are putting protein into small wells. There are 1536 wells on each of these plates. And then there are 1536 different conditions that may lead to crystallization. All of these wells then are photographed automatically by another robot, as you can see on the image on the left side, and the desired result is a protein crystal. But there are many different results that come from it. So the task really is to taking a large number of these images, which are characterized by many pixels, and try to extract some interesting, important morphological features from it that will help us to automatically identify what was the result of the experiment. One such example is highlighted here on a crystal, and from thousands of different pixel numbers, we would like to identify a smaller number of features that, when combined, can tell us if the result is clear, crystal, garbage, skin, precipitate, or a lot of the other different outcomes. The task is not simple. And it also required a huge amount of computing power to compute all of these morphological features. On the bottom side of the slide, you see just simple visualization of some of the results on a small fraction of these images, where with red we highlight clears, with green precipitate, and with blue crystal. You can also appreciate that there is mostly green, a lot of red, and you barely see any blue, which means that the crystallization, even with this wide range screen, is still a relatively rare event. Nevertheless, it's really complexity of this analysis starts with the size of the data. And we have been collecting this data over the decade. And on the right side, you can see, in a sense, the mass of this information. It's only about 60 terabytes of data, which by today's standard is not that big. But at the time when we were starting, a lot of the data was recorded on DVDs as a backup. And if you put all of the DVDs that have been created throughout this project, is 18-meter tall stack of DVDs, which is basically equivalent, roughly, to 4 London double-deckers put on top of each other. It's about 120 million images. The important part is that World Community Grid is a world effort, and the proteins for this effort come also from around the world, from over 1,000 different laboratories. You can also view it as a large number of millions of crystallization experiments. And importantly, the grid is the only window into understanding what was the result and how can we learn from all of these, in a sense, successes and failures of crystallization. Because less than 1% of these images have ever been classified by a human expert. This is not kind of our initial idea how to do it. We have been working on this problem for many years because it's not a simple problem, and we developed several versions of the algorithm. The cartoon on the upper right side highlights how we basically were going after. We initially identified a set of interesting characteristics from these images that correlated or related to some of the imaging outcomes from the experimentation. The problem was that as we added more features, we were able to improve the classification accuracy and thus ability to identify accurately what is the result of the experiment. But as we were adding more features, the program started to be intractable. We could not compute it easily on a simple machine. So the two things really happened. One was with the help of compiler optimization colleagues from IBM, we were able to improve the performance of the code itself significantly. But the second major aspect was that the World Community Grid and your donated resources enabled us to actually compute all of the results across these 120 million images in much shorter time. It would take us hundreds of years without the World Community Grid resources. So as a quick kind of highlight of this, initially we had a quick analysis which took about 40 seconds an image, but the comprehensive analysis that provided much more accurate results took about 2 hours per image. So you can imagine 120 million hours on a single computer would be required. With the optimization of the code, we were able to reduce the time only to 1 hour, but it still would take over 25 years on a cluster with 1,000 cores if we use it 100% of the time. First of all, it would not run for 25 years without interruption. And second of all, we did not have or do not have full access to this kind of a machine on a basis of 25 years. And also you can probably appreciate that no funding agency would be interested to wait for results of our research for 25 years. So we were able to short down the computation to about 5 years on World Community Grid. And as you may have noted, we also started graphical processing unit code on a World Community Grid. And the bar chart on the lower right side shows you how big speed up we can get on a different platform. So again, the interesting key message from this is that the technology enables new ways of looking at the scientific problem that otherwise would be prohibitively expensive. There are many approaches, as I mentioned, that we can improve the classification, that we can improve the algorithm for taking the features, and we are learning even from the data, obviously, that have been computed on a World Community Grid. Because what we need to identify is even faster way of computing these features now after we finish the World Community Grid phase. So as an example of where we are at the present time, one of the measures of how well these or how useful these features are that we are computing on a World Community Grid is to compare to human if we can classify images into the same categories as human experts would. And so we had about 200,000 images classified by 3 human experts, crystallographers. And then we identified how well we can identify clear, crystal, and other. And as you can see, the bigger the square on the left side of this diagram, the higher the accuracy, precision, and recall, meaning that we identify all of those images within that class and very few mistakes are made. The image on the right side is basically the same kind of visualization. The only difference is that now we try to predict multiple classes. And again, the important quality of the classifier and the features that we are extracting is that in a diagonal, you can see that these squares are relatively large. But also you can appreciate that for some images, we have a better precision or classification accuracy than for the others because some of them are more difficult, some of them are easier to identify. This we try to go back and forth because when we identify what is a true positive, meaning that we correctly identify what the human labeled as a specific type of an image, we can also learn from the mistakes because when we have false positives that we thought that it's a clear, for example, but they were crystals, we can try to fine-tune how come the algorithm made that mistake, what kind of features did we miss, and the same for false negatives. So it's really a learning process that enables us to incrementally improve performance of these algorithms. But one of the questions that now we can answer in a really global way is, humans gave us 10 different classes of images. Naturally speaking, are there more or fewer of these classes? So we started to look into, can we take these morphological features of images into consideration and how many different groups of images would we find? It's still very preliminary, but as you can see here, for example, we identified mostly different shapes of crystals. The other message from this slide is that what you are looking at is variety of crystals. So in one side, human says we are interested in crystal images. But you can appreciate that there is no easy way of specifying how does the crystal look because there are many different forms of it. Importantly for crystallographers or for the science, it's not as necessary to identify all of the crystals in the image. The important thing is to identify at least one of them if there is a crystal, because then crystallographers can optimize these conditions and grow the crystal large enough so they can do diffraction experiment, basically shooting X-rays through it, and based on the pattern of this diffraction, they can determine the structure of the protein. So these characteristics really highlight that 95% of the crystals will be in a top 100 images. So instead of looking through almost 10,000 images for any given protein, it's sufficient to look through top 100 and you will identify 95% of the crystals if they are there. The other way of looking into the magnitude of these results is that we look through about 1.5 million images that were never classified by human, and we identify what might be potentially interesting crystallization leads. Then through manual inspection of about 500 of them, because it takes a long time manually inspecting these images, we were able to confirm large number of these really being either microcrystals or crystals, although few false positives. So it already is showing that some of this data is incredibly useful because, as I mentioned earlier, only about 1% of these 120 million images have ever been seen by a human. The analysis will take a while. We finished the computation of the features for all of these 120 million images on a grid, but now we will be mining through it in many different ways using many different algorithms for foreseeable months. And the reason is that the data is incredibly dense and complex. What you are looking at is crystallization outcome is basically a large cube of data where protein information, we have proteins about 13,000 proteins. Then we have cocktail information, which is those crystallization conditions, and we have 1536 of them. And then we have 6 or 7 time points in average for each of the proteins. But each point within this cube is vector of about 15,000 features that characterizes the information computed on the grid. So as I mentioned earlier, one of the interesting questions is, can we find novel information in the groups of these images just by looking at their features and characteristics? Without going into too much detail, in this 9-way cluster, so identifying 9 groups of images, you can see that some of them are relatively homogeneous, although none is completely homogeneous. So there is a lot of heterogeneity that comes into these images. But for example, cluster 1 and cluster 5 are clearly identifying large fraction of junk, which are all sorts of problems with the crystallization. Interestingly enough, for example, cluster number 7 identifies large fraction of precipitates, and so on. So we will be able to extract additional information beyond what even human experts know. So in conclusion, really this reanalysis of 120 million images will identify novel crystallization outcomes. Through data mining of this large cube of information, we will be able to uncover some principles for crystallization and hopefully then improve speed by which we will be able to determine structure and function of biologically and disease-related proteins. As a first kind of preliminary statistics, it seems that we are able to identify about 60% of crystals in about 60% of images versus the current success rate for crystallization for high throughput screens about 47% and only about 35% in general. So we will still need to update these figures, but we should be able to significantly improve number of crystallization events identified. So I would like to really thank you all for providing resources to these projects and enabling us to compute and finalize this very interesting and important scientific problem. Obviously, I would like to thank Doctors George DeTita, Edward Snell, Joe Luft from Hauptmann Woodward Medical Research Institute, because those are the crystallographers that generated the data and provided for this analysis. Misho Kilimic is from IBM, and he helped us with optimizing the code significantly. And I would like to thank also World Community Grid and IBM team and users for their help and donated computing power. As I mentioned earlier, it's really a global effort, and there is every part of the world is producing results for this and many other World Community Grid projects. It's amazing that it basically computed over 113,000 years of computational power for the analysis. So the results and gratitude goes to all of you. Thank you very much.
Thank you, Dr. Jurisica, for this informative talk about the Help Conquer Cancer project and how our idle computer time is helping with the fight against cancer through worldcommunitygrid.org. We hope those listening are contributing and will encourage their friends to also contribute their spare computing power by joining worldcommunitygrid.org. We have time for a few questions. The first question is, can you go over again exactly what the definition of an image is in your project?
So every protein out of these 13,000 proteins is being subjected to large screen of different conditions that may lead to crystallization. There's 1536 of those conditions. Once about 200 nanoliters of the condition and 200 nanoliters of the protein stock are mixed together, then imaging setup with that camera that I showed on one of the slides is used for identifying all of the pixels, their different shades of gray. And it's about 400 pixels that we are analyzing for identifying the morphological characteristics of the crystallization. These images are not used for determining the structure, they are really used for determining which solution can lead to crystallization events.
Do you expect the project to need the resources of World Community Grid in the future again?
The main need for the World Community Grid was because we improved the algorithm for this feature extraction, also classification, and did not have enough resources to go and compute this backlog. On a day-to-day basis, and also based on the information that we learned from this computation from the grid, we have a code that we now are able to run on a day-to-day basis on our existing computing power. So for this project, we will be sufficient with our existing computing power. But as you will hear soon, we are starting a new project that is also related to cancer research.
Could you explain again a little bit why crystallization is needed and then the X-ray crystallography process that follows?
So as I try to put an example or analogy in terms of the Tetris, we need to understand the three-dimensional structure of the protein in order to understand how it functions, how it can bind to other proteins, how it can be affected by existing or potentially designing novel drugs to affect its function. There are many ways, for example, NMR, X-ray crystallography, and also computational methods to predict structure of the protein. But as I mentioned earlier, the most versatile method so far still is X-ray crystallography. The challenge is that those experiments have to start with a protein crystal, and that is rate limiting fact, because we might have a protein that we need to understand, but we may not be able to easily crystallize it. And that's the reason why this high throughput screen is essential for the project, because it can test thousands of conditions at once in very repeatable, robust conditions using automated robotic setup.
You described or mentioned that there are other techniques. For example, if you cannot get the proteins to crystallize, are there other techniques that can be used? And what are the benefits and limitations of the other techniques?
So as I mentioned, some of them, some of the techniques, for example, NMR, can be used on shorter proteins. There are methods, for example, that we also are trying to investigate where if the protein fails to crystallize by itself, one of the possible reasons might be that it needs its binding partner. And it's pretty much as a could be considered as a scaffold, that the protein cannot form the three-dimensional structure by its own. It needs its binding partner that, and afterwards it will be able to crystallize. So we use different methods for predicting what combination of proteins might need to be co-crystallized in order to help us determine the structure.
Is the Watson Research Center helping with your project in any way, or could it help in any way?
One of the, for example, interesting projects that started a while back, and there is being progress made, is, as I mentioned earlier, is a computational way of predicting structure. And the Blue Gene machine from IBM was one such machine that one of the problems, key problems, where this machine can be of help is to de novo determine three-dimensional structure. At the present time, still the accuracy and the throughput is not matching the crystallography. But as the question was earlier, for example, if we fail to crystallize a protein, there are other ways where we can get at least approximate structure. And it really will be combination of these different methods, and also what we learn from application of these different methods that in the future will help us determine structure of all of the relevant proteins.
What are the next immediate steps of your research, and do you expect to use local GPU processors perhaps to do some more computations?
So there are definitely several. One of the immediate aspects is, as I mentioned earlier, is that the features that we compute on a grid tell us a lot about what is important and what is not across 13,000 different proteins. So it's a great data set to start identifying what can we reduce or simplify without losing too much information. And it's definitely a first step, because we want to move this automated classification of crystallography images into the real time. Currently, the Hauptmann Woodward Medical Research Institute runs through about maybe 200 proteins on a monthly basis. So it's about 2 million images on a monthly basis. We need to be able to speed up the process of classification, so ideally researchers will be able to look through these images almost in real time. And so one way is to optimize the set of features, optimize the code, but the other option is that we will also run on a combination of CPU and GPU platforms, so we can run it on a lab scale as opposed to on a World Community Grid supercomputer. The other kind of follow-up task is, as I mentioned earlier, is to really analyze and mine all of this information that we computed from the grid and combine with the information we have about the protein, about the conditions, and identify what combinations of protein features and conditions for crystallization relate to the outcome, also considering the time scale. And so data mining, machine learning of this large set of data is going to occupy us for the next few months, at least.
Okay, is prostate cancer one of the types of cancers that's being investigated?
Many of the tools and many of the resources, and also this, is going to be useful for multiple cancers and actually also multiple diseases. But specifically in a lab and also with our collaborators, we focus on lung, ovarian, prostate. We started collaborations on head and neck, sarcoma, testicular cancer, and the list is really growing. So prostate cancer is also part of our efforts.
Do you have any guess about how long it will take for this science to turn into actual drugs and treatments and cures?
Um, as I mentioned on one of the slides, this protein crystallography project is only one of several that we are running in a lab. There is another arm of the lab that is focusing on computational ways of repositioning drugs. And the idea is to identify and analyze molecular profiles related to drugs. And we analyze molecular profiles from these multiple cancers that we focus on. And then we try to pair which combination of drugs is useful for which subset of patients with a given cancer. And we have now some preliminary results in ovarian and lung cancer where the predictions of novel drug combinations is actually better, at least in vitro, meaning testing it on cell lines at the present time, than currently available drugs. In ovarian cancer, we are also now running some additional experiments in a preclinical testing of some of these combinations. However, to say how many years it will take, it's hard to say because while these individual results are exciting, we have to test it in multiple different ways in different subsets before we can really start to be excited about moving into the clinical practice so we don't harm the patients.
Where on the web can someone go to find out about your future research related to this project and your progress?
I think on the first slide, I have a URL address that is a lab address, and links to those individual projects, to the publications, to the grants, to the updates on the World Community Grid are all under individual headings on that webpage.
I might also add that on worldcommunitygrid.org, we will post updates about your project on occasion when you publish papers or any other important things of relevance. Where do you get the proteins that you're testing? Are they from tumor cells or what?
So as I mentioned on one of the slides, the proteins really come from over 1,000 different laboratories around the world. Said that many are cancer related, but there are other proteins that are important for other diseases or are important for understanding how the healthy tissue and organs function. So it's really a combination of multiple biology and clinically relevant proteins.
Okay, would you have attempted this project without World Community Grid?
No, as I mentioned on one of those slides where I was highlighting the genesis of this computation, one of the revolutionary aspects of the research was that we were always obviously thinking of that we cannot compute things or functions that we cannot afford to do in a reasonable time. And it was kind of like chicken and egg problem because we knew that computing more functions will enable us better results for the classification, but that would require more time, which we didn't have. And so we were trying to go back and forth of trying to define of what subset of features extracted from these images is sufficient for a reasonable classification accuracy. And it was really the introduction of the grid. At the time when it was starting, it was only about 1 million devices, but still the computing power completely changed the questions that we could ask. Instead of trying to work with the limitation of the technology we had, we suddenly had the luxury of asking a question that made sense from a scientific viewpoint. How can we compute all of the features that we need to, and we don't need to worry too much about having available computing power to compute it? So it really revolutionized approach to the research or hypothesis and the question that we could form.
One more question. How long did it take you to develop the algorithms and code for this project?
So the answer kind of could be done in two ways. One is that the initial set of images or set of prototypes were created over the span of probably 7 years. Because as I mentioned, we were learning through the process of what is important, how can we improve it, and so on. But then once we had a reasonable prototype, it took about 3/4 of a year to prepare the program in its final form for World Community Grid computation.
Thanks again, Dr. Juriska, and those attending. Sorry if we didn't get to your question. Please use our forums at worldcommunitygrid.org to post any additional questions. This concludes our webcast.